Okay, 10 minutes, starting now.
All right, so my name is Toby, and together with Richard, Trinbin, and Mung, we work together on a fast semantic relationship browser. And this is a collaboration between TSRI, UCSD, and Star Informatics. Okay, so let me give a quick overview of the field at the moment. So at the moment, there's a lot of ways from going from primary literature, such as a PubMed paper, to a structured representation of that knowledge, which is usually stored in a big database. So you really have three ways of doing this. You have the human curators, the professional PhD people who read the papers and give you the structured information. You have a lot of extraction algorithms, such as SunRep, which do this for you. And finally, we're also working on ways which the crowd can do this process as well. But once the structured knowledge gets created, so far people haven't really found good ways of making downstream applications and what to do with those downstream applications. So in our lab, and for this project, we were really curious about how we can make a good browser for the structured knowledge database, and hopefully eventually how that will integrate into allowing us to improve the methods for which we extract this information in the first place, as well as how people can interact with this kind of information. So our goal is to A, provide users with the interface for this kind of information. B, and the actual primary goal is to allow these users to create and use our tool to create interesting and meaningful paths from that link concepts. So, you know, what is the genes that are associated with a specific disease, or what kind of drugs are related to this kind of phenotype. And then finally, as our long-term goal, we hope to eventually allow the users to contribute back into our knowledge database. So this ties into our lab's crowdsourcing activities. So potential uses, you can imagine doctors and scientists using our tool to formulate new hypotheses. So, you know, they see some patients in the lab, and then they can hopefully try to explain what's going on. And finally, maybe helping them discover things serendipitously. So unexpected things that they didn't expect to see that they saw with our tool. So this is a figure from a paper. So what happened is that this pathway of using GTP to synthesize neurotransmitters such as dopamine and serotonin is a very well-known and well-studied pathway. So what happened in this paper is that the doctors saw a patient come in with a disease that was a neurological disorder, and they didn't really know what to do with him because there were no drugs that treated that specific disorder. So what they did is they sequenced him, and they found that he had a mutation in the SPR gene, which is responsible for making one of these cofactors for the synthesis. And they discovered that once they knew that the SPR gene was mutated, they ended up giving the patient this 5-hydroxytryptophan precursor for serotonin, and they found that that actually had a positive effect on the patient's phenotype. So you can imagine that if we are able to produce these sorts of networks for everything in a patient or everything for biology, then suddenly it allows us to put things such as sequencing information into context, and this will allow us to make more rational decisions about what to do. So this is like the overall kind of database network that we hope to produce. Our method is actually, for what we did over the weekend, is we made a viewer for these kinds of networks. And the main advantages of our, which I'll give a demo later, is that A, it's fast. So a big problem is for any one concept, if it's a very popular concept, such as breast cancer, there's millions and millions of things around it. And we need the speed to display and actually prioritize these relationships, because no person is going to look through those millions of things by hand. You need some way, such as like to tell the person what's important and what's not, because we all know no one ever looks at like the third page of Google search results. And so we use an algorithm that's basically based on the Google normalized distance. So what this formula is telling you is that if two concepts are related somehow, so let's say they show up in the same abstracts of a PubMed paper, their relationship is going to be close if they show up together all the time, and they don't show up like separately. So concepts which are closely linked are those which happen to show up always together. So if they never show up together, then they're not going to be really important to each other. So now Trinbin's going to talk about what makes our database visualization system so fast.
Yeah. Actually, we come from UCSD database group. We had done some research on database for 7 years. And we found the bottleneck why database is not so fast in answer queries on big data. So we proposed our new algorithm and a new index, which can speed up the performance and also save the memory cost. As an example, we can save the memory cost from 38 gigabytes to just 4.7 gigabytes and speed up the performance by around 600 times. And the new index contains we use different complexion, and then we can load more data into the memory. And also we use column-oriented optimization, which is really achieving the high performance when answering queries. And we have a benchmark to show the result here. The running time. This is a low database called PostgreSQL. I think it used to be famous. And also a MongoDB, which is column database. And these two are our algorithm, which is really fast. And this is a space cost. And you can see we use less space and achieve the high performance. And we applied our algorithm in one demo here. So now to the demo.
Yeah, 5 more minutes, just so you know.
Yeah. Okay, so.
Can you make the website bigger?
Yeah.
Will I zoom in the view?
Yeah, you can adjust it.
Oh, yeah. I guess once you zoom out.
Okay, so if we go to home, so this is our interface at the start. So you start off with a search box, kind of like Google. So let's say you're interested in a specific gene. So I think for our demo, we're going to do T2P1. So you can search by the identifier for now. So this will bring up your gene, which is this TPP1. Right, and then so right now, TPP1 has hundreds, if not thousands, of things related to it. But we've already applied our algorithm to give you the top, I think it's like 10 things that are closest to it. So for here, we have things like late infantile neuroceroid leproscosis or something. So it's some sort of disease. We have malignant neoplasm of skin. So let's say you're interested in the link between this gene and the malignant neoplasm of skin. You can then click on the thing you're interested in, and it'll again apply the same algorithm and pop up other things that are closely related to this disease. So if you're interested in telomeres, you can click on that, and then in like 5 seconds, it'll show up with the new things. Right. So this will tell you that it's somehow related to DNA, that it's in people, and it's in chromosomes and aging. So at the moment, we haven't actually done the Google distance. We're actually using something called Jakarta distance. So the prioritization algorithm is a little different. But we hope to amend that in a later release. So that's our interface. And we're hoping to we didn't actually manage to get it done this weekend due to network issues. But we're hoping to integrate this with something that Richard has done prior to the hackathon, which is basically an online website where users can come and do this. So it's the same, it's very similar to this, and we'll bring it up.
So we've actually worked out that what we need to do to make the connection, but we just didn't have enough time to follow it up, and there were some practical issues with like servers and stuff.
It's extended. I can see that.
Yeah, so this will look familiar, right? So from here, we can again search for something like BRCA1. Right, so it'll pop up with a list of things that are containing the string BRCA1. So if you click on it, it'll give you a graph view. And let's say that we're interested in how BRCA1 affects ovarian carcinoma. We can drag and drop that relationship into the graph view. And then we can then look at all these 260 pages of results. And let's say we're interested in how Dutch population is the location of BRCA1. So again, you can drag that in, and you can move around your nodes and play with them. And the key thing here is that you can actually look at the primary evidence that this association came out of. So if we want to show why BRCA1 is associated with malignant neoplasms, you could click on the evidence button, and you'll see all the sentences from the PubMed papers that we got this relationship from. So here's a sentence, we can click on a sentence, and it should bring up a link to the PubMed article. So you get pulled up to the article and you can read it to your heart's content.
Also, the second tab there is the relation, other relations in that article.
Right, so for that article, this is a list of the things that we managed
Yeah,
to
there's
describe.
other semantic relations.
Right, so woman is the location of perpetuity. So this Richard came in having coded this before the hackathon, and we're working on integrating what you just saw with this interface, so that you can click a button and get a list of the important things around your concept without having to wade through 260 pages to find what you need.
This is a collaboration. I'm working with Ben Good and Andrew's larger team. We've also integrated another project called the Implicitome, which has a similar kind of. They're going to be publishing soon. So the second tab there gives you that, and you can drag and drop those things on. The second one is a certain one.
Okay, and you're just about out of time, so any parting thoughts?
Yeah, so in conclusion.
So what we're going to do between the two projects is when the first table, the explicit data comes up, it's actually going to go and harvest the high priority ones from there, and I'm going to mark them, highlight them, to be able, and even sort on them. So we'll be able to say these are the hot hits.
So for our future steps, we're going to work on improving the prioritization algorithm. So we would filter out things like human is the location of a gene, because everyone knows that. Or like, mouse is related to mammal. Next, we're going to, and we think one of the killer features is that we're going to integrate the ability for users to provide feedback. So if you look at the interface, you can imagine that when you ask for evidence of something, the user can read a sentence and say, I don't think this sentence supports the relationship that we got. And then they can say, contest this sentence. Or they could say, I'm an expert in my field, and I'm going to click on this button that allows me to add relationships, and I can give a list of PubMed articles that I think support my assertion. So that's what we're going to work on in the future, and we hope to launch this relatively soon. So that's about it. Thank you for listening.